Yeah, we all know that for the muscle contraction requires energy. Which compound do produces the energy? And we know that glucose itself is a simple compound which after decomposition produces energy. In this concise video, we're gonna deep dive into each and every step of the decomposition of the glucose, which is called the glycolysis process, and you can understand how different steps really helps in production of energy. And particularly when we start eating high carbs meal, what happens, our daily system gonna digest and produces the simple sugars like glucose. Glucose will be dumped into the blood. Whenever this blood along with the glucose passes over the pancreas, pancreatic beta cells will sense it. Pancreatic will sense it. Oh, this is the concentration of the glucose in the blood, so it can be dangerous. So what it does, it can start releasing the insulin. Insulin will be released in the bloodstream. This insulin will take the glucose along with itself towards the walls of the cells, particularly muscle cells and fat cells. And to allow the glucose into the cell, there are glycotransmitters which is present in particularly in the fats and the muscles. Without the assistance of the insulin, this glucose cannot be taken into the cell. So that's the reason why insulin is released into the bloodstream to push this glucose into the cell. Now, after this glucose enters to the cell, it undergoes different steps, or particularly 10 steps, where it produces the ATP molecules. Along with that, at last, it produces three carbon molecule called pyruvate. So now we can deep dive into that and understand few concepts of that, how this going to work. Imagine after eating, our digestive system has produced the sugar, which is glucose. And imagine this as a cell. We can take this as a muscle cell, or you can also take it as fat cell. Now I have already said that to take the glucose into the cell, this entrance of the cell has transporters, which is particularly blood-4 transporter in our muscles and fats. There are some other transporters, GLUT1, GLUT2, GLUT3, and GLUT4, GLUT5 also there. But these particularly when it comes to muscles and fat, GLUT4 transporter is present. This is insulin dependent. Insulin dependent. That means if the glucose has to enter into the cell, it has to come along with the insulin. Otherwise, this GLUT4 transporter does not allow the glucose enter into the cell. Now it comes here, so it is locked here. GLUT4 transporter does not allow. Now what it does? It comes along with the insulin. Now it comes along with the insulin, enters into the cell, and glucose molecule contains 6 carbon atoms, 12 hydrogen atoms, 6 oxygen atoms. So I'll just round up this one. This circle represents the number of carbons here. So we have 6 circles, 6 carbon atoms. And in the next step, what happens? One phosphate will be added to this molecule. So let's take with the incorporation of the enzyme hexokinase or glucokinase. Hexokinase is the enzyme present in the muscles and fats, whereas glucokinase is present in the liver. And it incorporates the ATP molecules. ATP molecules will break down to produce ADP molecules. In this process, it produces one phosphate. This phosphate will be added to the glucose. Now, what is ATP? ATP is the adenosine triphosphate. With the adenosine base, it contains 3 phosphates. Now, one phosphate bond will be broken, so that phosphate will be taken out and will be added here. And because it has lost one phosphate, it becomes diphosphate. Now, it also produces some energy. That energy will be utilized to add this phosphate into glucose. Now, what is the compound? This compound is called glucose 6 phosphate. Why 6? We can look at here. In this nomenclature, we can look at the carbon. This is a 6th carbon. For the 6th carbon, this phosphate is added. That's the reason why the naming itself is glucose 6 phosphate. Yeah, move to next step. In the next step, same number of carbons, same phosphate. So in the next step, it's just the isomerization. Isomerization is nothing but the change in the structure. So initially, in the previous step, we have the glucose. Glucose gonna be converted into fructose. It becomes fructose 6-phosphate with the assistance of the enzyme, which is phospho exos mutase. With this enzyme, this glucose 6-phosphate will be converted into fructose 6-phosphate. This step is just the isomerization. Let's move to next step. In the next step, what it does, let's bring in the enzyme. Enzyme is phosphofructokinase 1. Phosphofructokinase 1. This is very important step. This is irreversible step. And what it does, it's gonna take the ATP molecules, it's gonna break the ATP molecules, and ATP molecules will convert to ADP. Along with that, it produces the inorganic phosphate. This inorganic phosphate will be added to this fructose 6-phosphate, and that becomes fructose 1,6-bis phosphate. Yeah. In the next step, here at the middle, that bond will be broken and it will be separated into two different compounds. This compound is called dihydroxy acetone phosphate. Here, 3 carbon along with the phosphate. It is called glyceraldehyde 3-phosphate. And whatever the dihydroxyacetone phosphate is produced by breaking the bond between 3rd carbon and 4th carbon will again be converted to glyceraldehyde 3-phosphate with the enzyme phosphotriose isomerase. Now, this breakdown of 3rd carbon and 4th carbon is assisted by the enzyme aldolase. Yeah. Let's move to next step. In this step, how many glyceraldehyde 3-phosphate do we have right now? It is 2. From this step, from here it is 1. This is gonna be again converted to glyceraldehyde 3-phosphate. Right now we have 2 glyceraldehyde 3-phosphate, and here in this glyceraldehyde 3-phosphate will be converted into 1,3 bis phosphoglycerate. Where do this phosphate comes from? Here NAD positive comes here, takes away the hydrides as well as electrons. Along in that process, it's gonna add some inorganic phosphate to this, and this step is assisted by glyceraldehyde 3-phosphate dehydrogenase. So here we have 1,3 bis phosphoglycerate. In this next step, what happens? This phosphate will be ejected out from the first carbon. It just becomes 3-phosphoglycerate. This enzyme that helps here is phosphoglycerate mutase, phosphoglycerate kinase. Sorry. In this step, see, where do you think that this phosphate has gone? Now again ADP comes here. ADP is 2 phosphates are there with the adenine. It takes away this phosphate. This phosphate takes away this phosphate, and it becomes ATP. Now look at here. We have 1,3 bis phosphoglycerate. That means it has to 2 adenosine diphosphate comes here, forms 2 ATP molecules because we have 1,3 bis phosphate now. That's why. Next, move to next step. Here, no change in the no addition of any compounds or any molecules here, just isomerization. So here PO4 3 minus. Here phosphate is attached to 3rd carbon, and here it will be attached to 2 carbon, name will change, that's all. 2 phosphoglycerate. Enzyme that assists here is phosphoglycerate mutase. Phosphoglycerate mutase. Next step, same thing, nothing will change here, but compound itself will change, just isomerization, and this becomes PEP, phosphoenolpyruvate, with the assistance of enzyme enolase. In the next step, this phosphate will be broken and this phosphate will be added to ADP. Now again ADP comes here, takes away this phosphate, converts into ADP. We know that there are 2 PEPs, so 2 ADP forms 2 ADP. And in this step is assisted by pyruvate kinase. And this becomes pyruvate. Pyruvate is just looking at the 3 carbon. Okay. Now sometimes what happens, this pyruvate gonna convert into lactate with the incorporation of the lactate dehydrogenase. So for this step to happen, here we have produced NADH. So NADH comes here, drops the hydrides and electrons, becomes converted back to NAD positive. Two things. Yeah. So we can just analyze this once again. How many ATP molecules are required to assist these steps? Here we have 1 ATP, 1 ATP. So only we have supplied 2 ATP molecules to assist this entire step. So ATP used ATP are 2 ATP. How many in this entire process? How many ATP are produced? See here 2 and in this step 2. So totally 4 ATP are produced. And entirely, if you look at what is the net ATP produced, it is only 2 ATP molecules. Now these ATP molecules will, when they break the phosphate bond, becomes converted to ADP. In this process, energy is released. That energy will be utilized for muscle contraction. So this is how we get the energy from the glucose itself. And where do you think that this pyruvate go? So in presence of, if there is some oxygen here, so this enters into mitochondria. Where this pyruvate enters into mitochondria, there is a presence of oxygen and undergoes multiple steps, which enters into the Krebs cycle, undergoes multiple steps. Again, it produces NADHs, ATP molecules, or FADH2s. These things gonna produce. And where do these NADHs go? NADHs and FADHs drops their hydrides and electrons in the electron precipitation chain, and again in that process it is going to produce ATP molecules. In the upcoming videos, we're gonna going through that. Yeah, right now this is how in this 10 steps, glucose gonna be oxidized to produce 2 ATP molecules. But in these entire steps, there is no use of oxygen. This is very important, and I think why it is very important while you going to gym, why you have to take enough carbs, because to work out till the end of the session you need to have enough energy. So that's the reason why you need to take enough glucose. That means glucose is nothing but carbs. So this gonna produce enough energy. That energy can be used for muscle contraction, and you can intensely train the muscle group of muscles, and you can provide sufficient recovery, and thus the muscle become much more stronger after workout. So I think this one video has helped you to understand how glucose is important in the production of energy and muscle contraction. And if you find it useful, give us a thumbs up and follow my YouTube channel. Bye bye.